friends and donors and team. And I want to just introduce the team real quick and do a snapshot kind of where we are, and then we'll get into Rosa first, and we'll get into the lab, and then we'll do some back and forth, answer some questions, and get some of your feedback as well. So maybe we'll start with an intro. I'm Casey, founder and chairman of TCAR Rose Foundation, and I think most of you guys know that, so I won't go deeper into my theories or question. And then we also have Courtney, who's been our manager from the beginning. She's really been incredible. And let's see, it says my connection is unstable. So tell me if I start freezing up, just put it in the chat. Mulling has been the CSO off and on of TCAR, but also helped start the lab, and he'll get into more around what we're doing over there in the lab. But Rodney was the guy that the first lunch I had with the scientists way back in the day, and he left his very successful career in biotech to help these children. And I trust this man with my daughter's life. An incredible drug designer, drug hunter. Justin Fink has been a friend of mine since my 20s. He knows some of the things that I would never want to repeat about me, and he came in early with the lab at Everloom to help us learn how to create products out of what we were doing, so we could create some sustainability. Joe, I see you here all about the CRO. We had to create a CRO for families, and which is a contract research organization. And Justin, with all his sales experience, but also he's talked to hundreds of families at this point, and we feel very fortunate to have him running the lab. Lastly, Colleen is our new president, and very excited to have her on board. She's just been here since Colleen? Since February, I think. And has a direct connection with the struggles that families like her family and our families go, and these millions of families go through. Also an incredible leader and fundraiser, and is the reason why we're having a call. She's really great at communication, so this was her idea, and I think this is great, and I can't wait for you guys to meet her. So with that, I'd like to kind of jump in, back up a little bit, and go a little bit of give you an update on Rhodes real quick. And
yeah, frozen up.
Casey, you might need to restate what you just said. You froze. The joys of technology.
What about tell me?
Yeah, you're freezing up.
Freezing up.
But we have your audio now, Casey. We can hear you now.
Okay, I'm back. Can y'all hear?
He must be at his house in the hill country too.
Yeah, really.
Still unstable.
What about now?
That's better.
We heard that.
Better without your video, probably too much, enough bandwidth, I guess.
Yeah, we're on Musk's internet here. So I have another, I really only have 3 more minutes. I need to go about 3 minutes over, Colleen, on this, just because I want to, let's move to the next slide while I have connection here. So as we've been developing Rose's drug, our attempt has been to address this huge problem. And I think it's important to see how big this problem is. if you took everybody with these rare diseases, it would fill up 82% of the US population. At this point, we have a 1 in 20 chance of having a child born with one of these diseases. 72% of autism diagnoses are genetic. $1 trillion annual burden on the US. And we're discovering over 200 new rare genetic diseases a year. Next slide, please. The reason TCAR and other thousands of other family foundations are popping up all over the world is because our current model of developing drugs does not work for genetic disease, at least for 90% of them. And what we have been attempting to do is build a new model for how we develop these treatments and how we deploy them. You know, our past way of doing this has been a one-size-fits-all drug development, but with genetic disease, it doesn't work that way. We've got to target the underlying cause of the problem. Sometimes it can be one patient, sometimes it can be 50, sometimes it can be 500, but all of these diseases fall underneath the current sort of profitable model that pharma has. The other crazy thing is the technology exists to treat and even cure many of these diseases, but the business model doesn't. Next slide, please. So just this is what you guys have done. This is what we've done. And the title is actually wrong. I used this slide for something else. The title should be, we are a team, we are a lab, we are a new model. And you can see over the last 4 years, we started the foundation. We spun out Everloom Bio, which was a lab with $2 million of investment. We created a treatment for Rose in less than a year. Then we spun out Alpha Rose Therapeutics that the foundation owns equity in. It's a commercial company for these ultra rare diseases. We licensed in Rosafersen into the company, and I'll talk just a little bit more about that in a second. And now the investors got tired of us trying to squeeze our margins at Everloom. So we have the exciting news that we brought Everloom into TCAR. So Rodney and Netta and Justin at Everloom are now under the TCAR umbrella, serving patients and building genetic treatments for these diseases for families, working directly with families. Next slide, please. So the way we're reimagining drug development is through partnerships and through processes that work better for genetic treatments as opposed to these population-based medicines. We have the lab. We're able to have patient-led drug discovery here, which we showed with Rosafersen, but Rodney will show you with others. We're able to find diagnosed patients through genetic sequencing companies. We're working directly with NCAT to help these families through other scientific endeavors like small molecule repurposed drug screening and others. We work with Jackson Labs, who builds our animal models. And we work with manufacturers to create these, whether it's a gene therapy or an RNA drug, they're able to manufacture these, send them to us so we can test them. And now we have a commercial company that is, at least with ASOs, these RNA drugs, we're able to license these out either through the families or through the foundation into a commercial company to take it through the clinic and commercialize globally. Next slide. So why does this matter? And I'm going to start with Rosafersen. This is where I get really excited. It's because I'm hell-bent on sustainability. If I'm going to donate to a nonprofit, I want to know that they have some plan, that it's not some deep, dark black hole for the rest of the end of time without solving a problem. And so with our license, this is usually a slide I use to show investors now in the company. But I want to show you because your donations actually, we look at that as an investment in the foundation. And with our license, what that means is that when Rosafersen gets, say, a pediatric review voucher through the company, some of that money is going back to the foundation. When Rosafersen begins to create sales, that could be tens of millions of dollars back to the foundation through the licensing agreement that we have between the company and the foundation. And so my hope is that as we build more genetic treatments through the foundation that nobody cares about. We have companies like Alpha Rose, Mozzie, Amicus, Ultragenics, others that can license these treatments from us, take them through clinic and commercialize them and give us a return from the work that we did. So we're creating access, but we're also creating sustainability. Next slide, please. Oh, okay, good. That was it. So this was in short, sort of the vision that we're building this platform here through the foundation where we're able to spin out companies, we're able to create these pieces of the development path that are needed for a future of these small population drugs for these children, because the current system doesn't work. And I would say with through your donations, your support, and the brilliance of this team and our auxiliary team, our extensive team of partners, we've been able to get this far with this. And so, currently, last I'd say is that Rosaverse right now, you'll see a little bit of the science development, but we're raising capital on the company side to bring Rosaverse into the clinic and attempt to commercialize it within 3 or 4 years once we get it into a clinical trial. And commercializing means access in Japan, access in Europe, access in the US. And the plan is to give this drug to the kids that can't afford it. So in a ethical, moral, healthcare, biotech business model, you use your revenue to help support the underserved communities and even countries that can't afford your drugs. And that's how Genzyme did it, and that's how we're going to do it. So I'm excited about these partnerships we've created in the development path. And with that, I'd like to introduce Courtney, our project manager, that's been working so diligently to get Rosaverse ready for the clinic, and have her give us an update. Thanks. And I'm sorry I went over.
Hi, everybody. I'm Courtney. Like Casey mentioned, I'm the project manager at TCAR Rose. I've been there for about 3.5 years, and it has been a great experience, very fulfilling to work on this project. And I'm located in Dallas, and I feel excited and humbled to present this science to you today because this has all been possible because of your donation. So I hope you'll feel excitement as you see what your donations have been able to do for Rose and the other kids with this disease. So first, I want to start by just explaining her disease a little bit more. So it's called the HNRNP-H2 related disorder, and I'm just going to call it the H2 related disorder just to make it a little bit easier to say. So there are only 150 cases known worldwide, but the Broad Institute predicts that there are over 10,000 globally, and that these kids just haven't been diagnosed yet. They need genetic screening to be able to be diagnosed with this disease. And this disease manifests itself through developmental delay and intellectual disability, severe language impairment, motor problems, growth and musculoskeletal abnormalities, epilepsy. I know Rose has started having seizures in the last couple of years, facial dysmorphism, behavioral and psychiatric disorders such as anxiety, autism, and ADHD, and visual defects. We can go to the next slide. So I'm going to just describe this drug, Rosaverse, that we've created. You've heard the term antisense oligonucleotide before. This, I'll call it an ASO. So this is an ASO therapy. And let me just explain how these drugs work, and then I'll describe more about the one that we've created. So in healthy patients, we all have an H2 gene, and this gene turns into an RNA and then into a healthy protein. But in patients that have this H2 related disorder, they have a mutation in that gene. So one of those C's, G's, A's, or T's has been turned into a different the wrong C, G, A, or T. And that has caused, when that gene then turns into a protein, it becomes a toxic protein. And that's what causes this disease. So these ASOs are these really amazing little pieces of DNA that you can target to whichever gene in the body you want to get rid of. And it will prevent that gene from becoming a protein. And this is something that works temporarily. As long as the drug is in the body, you can get rid of that gene. So these patients would need to get this drug, have it be a recurring injection for them. But it's also safer. You're not completely altering the DNA. This is a temporary thing. And so the drug that we've designed, rosafersin, it targets that mutated H2 gene and prevents it from becoming that toxic protein. Thankfully, our bodies are very smart, and we have another gene in our body called H1 that is very similar to H2. And they found that in mice, if you can completely get rid of H2, that H1 protein will actually compensate for it. And these mice are able to be fully healthy. And so that's the idea behind this drug, is that if we can get rid of that toxic H2 gene, these patients, their H1 protein will start ramp up production and allow them to live a healthy life. So we started with over 55,000 computational designs for these ASOs. And we took the top 56 of these and we screened them to make sure that they worked in human neurons on a cell culture plate. We took the top 7 candidates that looks like they were decreasing that toxic H2 and increasing H1. And we tested those for safety and for efficacy in mice. We wanted to see that they were effective at eliminating H2. We took the top 2 candidates from those mouse studies, and we have just tested them in rats to make sure that that drug is safe in an animal model at a high dosage. And so we recently got those results back. We have 2 candidates for this drug that look really good and safe in this animal model. And we will now turn these final safety studies over to Alpha Rose Therapeutics, the commercial company that Casey was talking about. And they will then finish up the last safety studies. We'll do it in rats and then move to a clinical trial and test our top rosafersin candidate in a clinical trial. And we can go to the next slide. So there are a few other areas we've been working on as well simultaneously. So one is drug repurposing, and that is exactly what it sounds like. We take a drug that has already been developed, already approved by the FDA, and see if it improves any symptoms of this disease. So we are currently looking at a whole, a number of drugs that target epilepsy and drugs that are already known to decrease H2 in the body, and we're going to test them in cells from patients with this H2 related disorder and see if any of them improve symptoms or decrease H2 and increase that H1, because that's an easier way to help relieve symptoms. And then the other way is through biomarkers. So biomarkers are like little tags on DNA that tell us about the health of the cells. And so you can take a patient's blood and look at the DNA in the blood and see what markers they have on their blood. And so we're finding which markers show that they have this H2 related disorder. And then when we treat with our drugs, we'll be able to see, do the markers change? Do they look more like a healthy piece of DNA with healthy markers on them? And next slide, please. And most recently, we have been organizing our natural history study and clinical trial. And we've started with a platform at Citizen Health. So anyone who's interested in joining this clinical trial or a natural history study can apply at Citizen Health, and they'll collect all of their medical records so far and have them all in one place. And then we have a few different arms to our natural history study. So one is with a company called CISNAV. And what they do is they have a little wearable that the patients will wear on their ankle, and it'll measure their walking, their gait, basically their motor function. And Rose has tried this already. She was our first participant in using the CISNAV wearable. And so it tracks their movements, and then we can monitor that over time as we start our clinical trial. So the amazing part about this is that these kids can be their own control in this clinical trial, because we will monitor their individual metrics as we treat with the drug. Another arm is we are creating a video assessment tool. where the caregivers of these patients will be given a list of tasks. Could be simple tasks like picking up a fork or saying a certain word or maintaining eye contact. And the caregiver will video that task, and then we can monitor those tasks over time as well to see if our drug is improving these daily life tasks that these patients do. We've also partnered with Simon Searchlight. They already have a registry full of patients with this H2 disease who have donated biosamples there. They will help us contact the patients and help them get them registered for the natural history study. And then we have partnered with Colorado Children's Hospital, and that is where we will run our clinical study, where these patients will receive the drug, and where Rose has already been there. She went there, visited in March. They get patients get a full evaluation by a pediatric neurologist, and they'll measure, all of their metrics and chart them throughout this study. And you can go to the next slide. Thank you, Colleen. And so, yeah, thank you for listening. That is our progress so far. We are excited to be so close to a clinical study. Hopefully we'll be able to start that trial in the next year. And just thank you for your donations and making this possible for us to develop this drug. And I will turn it over to Justin.
Okay, thank you, Courtney. That's incredible work. Hi, everyone. I'm Justin Fink. I am the president of the new preclinical TCAR Rose Rare Disease Lab. And I want to take a few minutes to share not only what we're building, but why it matters so deeply and why I'm glad to have you join us here today. When a child or an adult patient, for that matter, is diagnosed with a rare disease, time becomes the most precious and terrifying thing. We call this the time is life paradigm. Unfortunately, the traditional system, pharma pipelines, clinical research organizations, academic labs are not always built. There are some, we've worked with some, but they're not always built to respond to families racing the clock. That process is a little too slow, too siloed, too expensive. And that's where our lab was born, out of urgency, necessity, and a demand for speed, and most of all, great science. When Rosie was diagnosed, Casey, like many others, found that the same old existing system. So he envisioned a lab for rare patients, and we set off to build one. Alongside our Chief Science Officer, Rodney Bowling, and supported by an investor and CEO at the time, we created a new model at Everloom Bio in Casey's vision. A lab where families can own their data and IP, our science team can move quickly, and pursue multiple therapeutic paths at once. We call this the multiple shots on goal, a modality agnostic approach where science leads and gives the patients the best chance towards a treatment. In January of this past year, the CEO of Everloom abruptly in a meeting decided he no longer wanted to support the lab, nor did the investors. So in February, Casey, myself, and Peter, along with the support of the TCAR Rose board, created a plan in record time for the lab and the team, as well as the patients we were working with, to go from Everloom Bio into TCAR, providing patients the only preclinical rare disease lab in the world that resides inside a nonprofit. In just 2.5 years, this lab team has delivered 3 successful preclinical therapeutic programs. As Casey mentioned before, we've spoken to over 100 families and foundations. And currently today in the lab, we have 13 active programs from patients around the globe, from Australia, Austria, Singapore, the Netherlands. This truly is the start of a global lab. And as an Austinite, I am proud to say that it sits here in Austin, Texas. The lab is truly a place of true partnership between families, patients, scientists, and collaborators, and we are just getting started. In rare, most, if not all, families are often self-funding. They're met with not a lot of answers. There's not a lot of market incentive from traditional industry to help their loved ones. And with this lab, we are saying we're here, we're coming, and we're bringing science with heart. Every family we work with becomes part of our family. Every breakthrough, every setback, we feel it. We meet with them monthly. This is personal because this is truly personalized medicine. As we move into a nonprofit, I came across a quote by Seth Godin. I'm not sure if you've ever read Purple Cow. He's a marketer, a great speaker. And it read, "People like us do things like this." And it just really struck me at the core. It kind of really started to make sense about this transition into a nonprofit. And our entire team and folks like yourselves giving some of your time today, that's truly what we're all about. People like us do things like this. Transition the slide, please. Okay, on the left here is a photo of an 8-year-old girl named Sasha. And I want to share an email with you now. And I'm going to get a little emotional possibly, so bear with me. Sent to me on April of this year, and just gives you a little peek of a window into the days of a day in our life, and further displays the reason why we exist and we need support. And it reads, "Hi, Justin. I saw a post on Facebook for TCAR Rose and Rare Labs. We are in Australia. Sasha, my daughter, has a splice site mutation." And you say that 3 times, right? That we believe is treatable with an ASO. Can your lab help us? We have been let down by groups here in Australia in the past. Sasha is extremely affected by her disease and has regressed. This is Sashi. She is an absolute angel, purest heart and soul. For Sasha, we believe a personalized ASO is the only hope we have because of the time. And we keep hearing that time. And the fact that her mutation is most suited for an ASO. Dave and I will do absolutely anything to help Sasha. She can no longer talk, and she has lost every single skill she once had. She is 8, looking forward to hearing back. Nadine. Now that kind of message hits me hard every month that I see them, but it also serves as a driving force for us. It reminds me on a daily basis to go downstairs and hug my 2 healthy kids. And I know that not every parent gets that kind of time or moment. To the right, though, is just 2 weeks ago. And this is personalized medicine. This is our science team meeting with the parents and their science team in Queensland, Australia. Starting to collaborate and seeing if we can provide a potential proof of concept treatment for these parents. The lab is located at the Austin Community College Bioincubator space. It's a wonderful space. There's tons of room for growth, and we have a highly collaborative vision with the folks there, which is very exciting. You know, we just met with them 2 days ago as well, and we'll be meeting with their executive team shortly. I'd like to personally invite each of you, if you're ever in Austin or if you reside in Austin, to come on over to the lab. You can tour the space, talk to myself, talk to the team, meet the scientists. There's a great little coffee shop right off of campus, and we'd love to just sit down, get your feedback, talk about our vision. And I can put my name, obviously my email, and my number here in the chat. Feel free to contact me anytime, any day, maybe other than Sundays, but we're working around the clock here. I'd like to conclude by saying that this truly is hard work. This is personalized science and medicine. It's emotional work, and rare disease obviously is a massive problem worth the attempt to solve. And it's why it's in TCAR Rose, because it's truly why people like us do things like this. And so I want to thank each of you for your time this afternoon. And now I'd love to introduce someone who's been at the heart of this from day one, my brethren, my colleague, our Chief Science Officer, Dr. Rodney Bowling.
Thank you all for being here. Hearing Casey's emotions, Justin's emotions, thinking back on these kids, I'm fighting back tears myself. This is truly an amazing team and truly an amazing opportunity to not only continue helping Rosie, but continue the mission of helping all of these children. I've been in the drug discovery game for 18 years. I have 17 drugs currently in clinical trials. Hopefully soon 18 with Rosie's. We have adopted a modality agnostic approach. What does that mean? We work with antisense oligos, which is a little piece of DNA or RNA that we inject into the child, and it can work in different ways to cause more protein to be made or less protein to be made. It all started with Rosie here on the left. Here in the middle, behind these beautiful blue eyes, this poor woman's brain is dying a little bit every day. In her case, we have a gene therapy that delivers a working copy of her SPG11 gene into every cell in her brain, hopefully. And this would restore proper function. You can see her cell line down below. The blue are the nuclei, the red is her disease. When we add this virus, the lysosomal storage disease shown in red, just 3 days after the virus is completely gone. Going to take this opportunity to call out Netta. She's not on this call today, but it's her hands that have done all this work. Finally, our last modality, this little guy over here on the right, this picture was taken 3 years ago. Within the next year, he won't be able to walk. He will lose all of his words, and he will be confined in his bed for what's left of his life. Sanfilippo B is a crippling disease that ends your life sometime in your second decade, but functionally ends your life after your first. We have found through a high throughput small molecule screen of all things an antibiotic that will shut off his lysosomal storage disorder and hopefully heal his brain and give him a little bit more time. All of these studies have been funded by the parents of these kids. And what we have found almost universally is parents with sick kids have spent all the money they have taking care of their sick kids. We hope to change that with Rosie's drug. Like Casey said, there are potentially 10,000 children out there that could benefit from the work that we've done, that Casey's done with Rosie's drug. SPG11, there are probably another 5,000 people. We currently are working with 4 families in the United States and a large group in United Arab Emirates that all have this SPG11. And Sanfilippo B is not so small. That's as many as 50,000 patients in the world that could benefit from this. This is a snapshot. These are 3 patients of the 13 that we've worked with. We've had some more exciting news recently. I very much appreciate your support of this work and the way that your support blesses these children. Thank you very much for your time today.
Okay, now you got me tearing up. Wow. Okay, I will keep this short and sweet before we take a few questions. First, I really just want to thank our team. Thank you, Casey, Courtney, Justin, Rodney. I mean, the work that you do each and every day, it's truly inspirational. You're all my heroes. For those of you who don't know me yet, I'm Colleen Anderson Hawthorne. I'm the new president of our foundation. And as Casey had mentioned earlier, I am a mother of 2 daughters with living with a chronic disease. And this mission, it lives close to my heart. I know what it's like to live in this space between fear and hope. I know what it's like to be on the receiving end, waiting for a treatment and a cure. That's why today matters. What you've seen and heard isn't just promising, this is transformational. The science that we're advancing is really changing the course for these families, for these children like Rosie, for everyone who's impacted by these devastating rare diseases. Your presence here today and your willingness to be a part of this movement, it just means everything. And I know some of you have been here since the start 4 years ago. Some of you have just joined us recently, but we can't do it without you. So on behalf of TCAR Rose, thank you. Thank you for climbing with us and fueling our mission. In the next few months, we will be launching a bold new 3-year campaign that we're calling Rise for Rare. And if you're ready to take the next step with us and rise with us, I'd love to keep this conversation going. For those who reside in Texas, very excited. I will be going to Austin for the first time this June. I reside in Jacksonville, Florida, and I would love to connect with you all in person. As Justin and Rodney both shared, our team would be honored to walk you through our incredible rare lab and show how your support is bringing real hope to Rose and other families you heard about today. So thank you for joining us and for being a critical part of this journey. Okay, I know we allotted some time for some questions, so I'd like, if you have a question, please feel free to unmute yourself, and our team is here to help answer some. Who would like to kick it off?
Barbara always has something up her sleeve. Well, you're muted, Barbara. I know your wheels are always turning. So
My
I'm
wheels
going to
are.
call you out first.
Well, I'm a little confused. I've heard this story a couple of times, but just doesn't seem to gel. You started off with TCAR Rose, which is a nonprofit, right?
Mm-hmm. Right.
Then you got the lab started, and you made that a biotech, whatever the name is, Illume.
Yep. Company. Everloom.
Everloom, yeah, Everloom. Okay. Then you've got a drug called
Rosafersen.
Irricin or something.
Mm-hmm.
So you've licensed the drug, but you made the drug. So why do you have to license the drug? And I mean, I got that Everloom is now back with the nonprofit. But I don't, let's see. The profits from the drug that you made, you're going to split and put some back to the foundation. Is that the point?
Yeah, I can explain the structure a little bit better, Barbara. My bad internet kept me from going into more detail.
Yeah.
So, the current structure with drug development is that academics typically make a proof of concept therapy. They're like a nonprofit organization, supposedly, but they have a tech transfer office full of lawyers that create licenses for biotech companies to take those proof of concepts through the clinic, because then you're looking at, for a typical drug, $500 million to $1 billion in investment, right? And so for these rare diseases, we're thinking it's going to be more 15 to 30 million to commercialize these treatments. If they're under, say, 1,000 patients, the more patients you have, the more expensive the clinical trial is going to be. And so in some ways, I'm adopting that model, but the difference here, so TCAR, the foundation, and the lab, which the lab and the foundation are the same thing, right? So it's all a nonprofit. That becomes our place to do preclinical work, which no one will invest in. And it's actually not that investable because you don't have any intellectual property. And then it also becomes a place to do our biomarkers and our natural history study, which could never be owned by a company because investors don't really like you to share data. And biomarkers and natural history study need to be in a place where data can be shared. So if there's some other company that wants to make a better drug, they can go back to TCAR and say, hey, we want some of your, natural history study biomarker data. So the reason we spun out the commercial company Alpha Rose is the idea that I could raise $30 million in donations was just impractical. Plus, if we keep, I really think sustainability is key for these medicines. So meaning like I've been I've been on boards of nonprofits for, I don't know, decades at this point. And one of the things I hate is when a nonprofit is constantly doing stuff that requires more and more donations without a plan to create some level of sustainability in fixing the problem. And so a nonprofit's not perfect for raising investment money, but it's perfect for raising donations around preclinical proof of concept, things that should be in the nonprofit. But then how do we make it sustainable? Well, we can make it sustainable with profit, but that shouldn't be a nonprofit's goal, and nonprofits get to be pure mission. So decided to spin out Alpha Rose, late 2023, we capitalized at 2024. It's a public benefit corporation. So it was the best I could do from a corporation structure. And what that means, this blows my fucking mind, is that all our healthcare systems are corporations that have one single goal: fiduciary responsibility to shareholders. That's it. No responsibility to patients, no responsibility to the people they serve. So the only structure that I could find that helped us create an ethical model was a public benefit corporation, which means we have a fiduciary responsibility to our shareholders, but we also have a fiduciary responsibility to our customers, which are patients. And so it allows me to put a moat around the investors. So they're not just going for the thing that's going to get the most amount of revenue, but it be a profitable company that's good for patients, right? So like an ethical company. So the foundation owns 5% of the company. The license requires milestone payments. So the foundation licensed the intellectual property of the drug to the company. So the foundation gets milestone payments, and then it gets a royalty on any revenue. And all of those numbers are standard, what a typical, that I wanted to be careful, so our lawyers made them a very standard licensing agreement that looks normal. But there's also a very important provision here, which is clawback provisions. So if Alpha Rose goes under, something bad happens, or Alpha Rose decides to not develop, my investors, I do it wrong, and investors do like every other biotech company where they tell you to shelve the drug, which is why we didn't license it to someone else, TCAR has the ability to get that drug back. So I think from an ethical standpoint, I'm really happy with the license. And I think where we have to charge families for some of this drug development, and I think that's where Justin and Rodney really struggle is what is the model for the lab going forward? Right now it's sustainable and that it becomes a lab for families to do this drug development where they don't have to become scientists or project managers, but
Lost you again. Okay. So
Sorry.
can everybody hear
I'll
me?
stop talking. Yes.
So that was helpful. That's helpful. So the company is supposed to make money. Some of that money goes back to the foundation so you can continue to do research. And some of the money goes to shareholders as profit when the drugs become successful and are sold or whatever.
Correct.
That's called equity something or other. What is that called? Equity growth? I've heard the term a couple of times. It doesn't matter.
In terms of, yeah. Yeah,
Like-
so I mean, it'll be revenue from the drugs, and then shareholders will make money off of their stock that they have in the company. And so we plan on going public with that company instead of selling it to a big farm. We'll
Right.
PO that company in 5 to 7
Well,
years.
Okay, I understand that now, and I just want to compliment the lab, not just Rodney, because I know he's head of the lab, but he's got Netta and whoever else is in there. You've come up with 3 successful treatments in 4 years. I mean, that's pretty good, right? I'm just working on one. And I've been at it for 4 years too, and I don't have any. So congratulations on that. Absolutely.
Thank you, Barbara.
You're welcome.
Can I say one thing?
Renee, yes,
Hey.
please.
I just wanted to say thank you all for this whole presentation, because it was very informative and actually needed, I felt like. And then I want to say to Barbara's question, I'm on the opposite side of that. Casey's heard me say, and other people, probably several other people that have been with him from the early days, is that when this goes to a drug, that I would like to see a close the loop back to the foundation, so that the ethical side of the drug industry, which doesn't exist in many places, it's all a profit center, which is great. You know, we all want to make money, but in the big pharma industry, it's a profit center, and the ethical side of it kind of goes to the wayside. I love that you guys, whether it was out of necessity or not, finally closed the loop to come back, so that you have a something not only symbiotic, but something that balances your potential cure and profit center, and closes the loop to come back to the ethical side of it. So I think it's awesome.
Good.
Thanks, Renee. Christy, it's good to see you again. I don't know, you know, we haven't talked in a while. I'm curious how, you know, you can't ask this is a very unique thing and way that we're approaching this too. And so I would love to hear your feedback and any questions that popped into your head.
Well, hi, good to see you. I fully agree with Renee. Renee just said it beautifully. I haven't been following the details. I've been following my heart just to support you guys. But yeah, I've teared up today many times through this, and it's just it's amazing what you've done. I think you guys should all be proud of how much how much success you have from your hard work. And you've come, even though it's been I'm sure it feels very long for you, you guys really have accomplished a lot in a short period of time. And I think it's amazing. So I'm glad I had the opportunity to hear this. And thank you for spending the time, probably repeating a lot of things that you talk about every day to all of us, because I think it's well received for sure. Did I go out?
No, thank you.
Oh,
I
sure.
can hear you. Thank you for that.
Yeah, sure. I think Casey's gone again, isn't he?
And thank you, Renee. And thank you, Barbara, for asking a very critical question too. Yeah, I think Casey's coming in and out. Does anyone else have any other thoughts, comments, questions for us?
Colleen, this is Joe McCafferty. I have a question. Just Casey and I hooked up very early in this process, probably 5 or 6 years ago, and I'm really impressed with the progress that the lab has made and the overall foundation. Question I just have, because I haven't been close to the progress of the different programs over the last year or so, but is H2 still the lead program? And if so, where does the SPG 11 and the Sanfilippo B program fit in a continuum? it's impressive that you have 3 programs progressing at the same time, but I'm just curious, how are they comparing in terms of how they're progressing towards whatever the next milestone is to the clinic for H2 with Rosafersin, or with the others, the gene therapy and the small molecule?
That's a great question, Joe. I'm going to let our Chief Scientific Officer answer that because I feel like he would be the best one.
Sure. Let me say one thing first before Rodney gets in, is that there's 2 big differences, Joe, is the H2 program is controlled by the foundation, owns the intellectual property. The services from the lab standpoint for the families, one of the big things we do is we say, look, if you're paying for this, you own it, which is different than academia. Right. So they own all the intellectual property. And it's one of their challenges now that Justin and Rodney are facing, is when we get to proof of concept, are there any CROs, sort of clinical development teams that we can move this into? Because we don't have the capital or the expertise to move them into the clinic. And so now that that's something Justin and Rodney are working really hard on, and us as a team discussing right now, of our best work is done on this preclinical side, but who can we partner with? How can we hand them off to somebody? Eventually, we'd love to do all of it, but right now we don't have the capacity to do it.
Sure. Understandable.
Rodney, Justin, you have anything to add to that?
Yeah, I was just going to say, just to be clear, so in those 3 examples, obviously, as we mentioned, there's 12 programs. These are services, right, that these families are self-funding, or i.e., in the case of like TCAR Rose with Casey, right? They have their own foundation, they raise money and pay for services. So the collective vision from, when we look at this is, are we able to continue that track and that path today with the work that's being done for Rosie and those patients, but then look at a broader vision around subsets of different diseases, raising donations for the collective, because one of the missions in preclinical is, can we reduce the costs but keep the time, and this approach and this kind of new approach of personalized medicine and rare disease for as many as the lab can handle at any one given time. So I hope that provides some clarity on your question there.
It did a little bit. Thank you. Thanks, Rodney. Or Justin, sorry.
Mohammed and Andrew. I see Andrew's got his hand up, and you guys are thanks for going. You're coming visual. So maybe Andrew will take you with your hand up, and then Mohammed.
Sure.
I got extra
thanks.
time too.
Thanks, Casey. Yeah, for the question, James, really personal question for you is sort of, it sounded like from what I could gather, I'm sort of an outsider looking in, that the drug isn't ready for Rose to actually try the drug yet. And I know it's a personal question. If you don't want to get into it, you don't have to. And when will that happen, or has she tried it? And, what's the update on that?
Yeah, so with these types of diseases, they're neurological, right? And so they require the drug to get into the brain, which right now requires an intrathecal injection, which is an injection in the spine. And so, step 1 was to make sure it's safe. At this point with our studies, we're confident that it's ready for a clinical trial. Step 2 is making sure the FDA is ready for us to do a clinical trial. So we still, the IND enabling safety study is $700,000 or $800,000. The same study we just did with rats for $100,000 that is not approvable by the FDA. I'm gonna have to do the study again for $700,000 because of all the paperwork and check boxes that are required by regulatory. Pretty much same study. And so, I'm trying to raise that money in Alpharos and the company. And once we get that done, our regulatory teams are set up. So we should be able to treat kids Rhodes and, whoever else is admitted into the trial, I would guess anywhere between 6 and 10 months from the point that I've got the capital to sort of like flip the switch on with manufacturing, fill and finish, IND enabling studies, and the regulatory team to write the IND.
And you need some funds to be able to do that, or it's just time?
It's funds right now, so that's on the capital side. So we didn't want to put that sort of those types of numbers on the foundation, So that will be through equity in the company.
Thank you.
Yeah, thanks, Andrew. Great question. Mohammed, great to meet you.
KCI, as you know, I'm a scientific officer with Barbara. And since I joined, I heard about you a lot. And then I had direct contact with Rodney and Justin. Okay. But I never met you, and I never met any of you directly, at least face to face in a video. But it is great to see all of this moving forward. It is really, I did not know you have achieved so much. You said now 13 program. Okay, that is a great. Okay, and then I have been hearing from Barbara since day one you started your lab in Austin. Okay, that Rare Disease Center. And I said, man, that is a great thing. And you are working in the lab, your people working in the lab, doing clinical work. Because I know I am the like a people most of my life worked with the preclinical aspects. You know what I mean? And now thing is, in your clinical lab, are you doing any cell culture work or just like a natural history and things like that? Are you planning to do some cell culture work and gene therapy work and things like that, or just ASO?
So our lab, we start by collecting a cell line from the child, and we call this our toolbox phase.
Okay.
We take and make an inducible pluripotent stem cell from that child. And then depending on where the disease is, and most of these diseases they're honestly in the brain, we differentiate neurons, and we test our treatments where in the context of the disease in these patient-derived neurons. And then through sequencing, assay development, we look to find which modality is going to be most appropriate. If it's an ASO, we go that route. If it's a gene therapy, we go that route. And if it is a small molecule, we go that route. And we try to take multiple shots on goal, depending on timing and what the parents' finances allow.
Oh man, it's
Mohammed,
yes.
We got that. So one of the things I loved when I met the team at Ultragenics was that they were a multimodality company. So they weren't specializing in a particular technology. They were thinking, what's best for the disease? And then, and most companies actually don't operate that way. Even Alpha Rose is specialized in commercializing ASOs because they're ready for these small population diseases where gene therapy is a little bit more difficult with the small population. And so one of the things I think that I really commend Rodney and Netta in is they establish enough knowledge in multiple modalities, but they have a network of experts that Rodney's calling on, depending on if it's an AAV9 or Lenti, so it's an repurposed drug. So
freezing.
You froze up again, Casey.
I think so. Anyway, that's the modality approach that we really are proud of.
Yeah, I really got that idea because most of the crucial proof of concept, feasibility, those are done in the lab. and you determine actually which way the disease goes. Okay, what can be best for this, at least initially. Okay, and that is the real thing. You know what I mean? That is the real thing. Okay, I really appreciate all of you. I mean, knowing that your success. Okay, thank you so much. Okay, and thank you for the opportunity. Okay.
Thank you, Mohammed.
You're welcome to come into the lab, and
No, I
Rodney's
would.
always
Okay,
looking for help.
because I might visit Austin, and then definitely I'll go. Okay.
Fantastic. All right. Well, anybody, I think we've come up, we've gone a few minutes over, so I apologize. If there's any other burning questions. Craig, you've been quiet. You got anything on your mind?
Yeah, well, I want to say congratulations for all the progress. It's just amazing. You know, we've been with you for since COVID started, and
Yeah,
it's just the accomplishments that you've achieved are just amazing. So I want to just say thank you for sharing all that information about it. It's amazing to see what you've done. One thing that I was thinking about as you've gone so far now is. Do you see any obstacles with the current administration in Washington and the FDA getting in your way as opposed to the way it used to be? Do you think that's a problem?
It's a really great, interesting question that we're all talking about right now. So fundamentally, rare disease is not a partisan issue, right? Thank God. As long as we don't make it one. And so, I was up on the Hill a few weeks ago talking to congressmen and women on both sides of the fence. What's been interesting is there's actually a lot of hope that we can make. So we have too much regulation, and we have a sort of calcified regulation that feels good for big drugs. We've like FDA's made sure that drugs were safe, and in the 40s, 50s, 60s has been but now with genetic testing, with these thousands of new diseases, we know that things have to change. So there has been some what would you call it, taking regulatory apart right now, and which is sort of scary. So it could end up that we're totally screwed and nobody cares about it, and we're set back a decade. What is more likely to happen? Had of the like roses.
Again.
So I'll go backwards. McCary, can y'all hear me okay? McCary has mentioned diseases like roses and that it needs a new clinical path. So I think we actually have an opportunity to make things better in this administration.
Good.
Yeah. Yeah.
But we'll see. All right. I know we've gone over, and I want to respect everybody's time. So I appreciate all of y'all's involvement and continued support. Like we've all done this together, which is really quite incredible, So thank you, Colleen. I don't know if you have any last words.
Yeah, just thank you so much. As I started in my new role here a couple months ago, I know I've had the chance to talk to a few of you on the phone, and again, I'm really looking forward to meet some of you in person, if possible. But thank you for moving our mission forward. Like I said, we can't do it without you. So sincere gratitude.
Reach out anytime. We'll talk to you guys soon. Thanks for being